So in this video, I'm going to talk about signal transduction and the endocrine system. And again, just focusing on the big ideas of this process. So in this video, so what I want to first talk about is the this general signal transduction pathway, right, which takes place inside inside the cell. Now, I know I mentioned the endocrine system here. That is a system of your body, and in every organism, right? In humans, it's within the pancreas that sends out little messengers. It sends out signals when it wants the body to do things, right? And the signals reach the cell, and now we want to look at how does, when a cell gets a signal, right, it gets in the form of a signal molecule, which is called a ligand, and I want to know how do you go from the signal to a response in the cell, right? And so what's going to happen in general is you have a signal, it binds to a protein receptor in the cell. That protein is going to activate a molecule. Then you have what's called a phosphorylation cascade. And what that is it's going to require energy, right? But again, this is all kicked off from this signal. You are going to have a phosphorylation cascade where you have ATP is going to react, and if you want to learn more about ATP, I'm going to have a video about energy-coupled reactions soon. But essentially, you know, it's going to add energy. It's going to add this phosphate group, which is going to increase the energy of the protein, and that will activate the protein, right? It's and so, you know, if the protein is supposed to look like this to work, right? Before it has the phosphate, it could look, you know, all crumbled up, and, you know, and then you add the phosphate. Go ATP goes to ADP plus PI, right? And you have a phosphate here. It gives it energy. And the protein kind of goes through a conformational change. You kind of see it's the shape change here. And it's going to change form and be able to be active, right? And then it's going to do something. It's going to relay until it gets to the certain protein that it activates, and that protein is going to carry out the cellular response. So in essence, right, I want to look at the big pictures. So structure and function, right? What does this do? Well, the protein structure is important. The membrane makes it so you have, right, the structure of the membrane allows for you to have this protein there, and which will allow for signaling, specialized signaling in this pathway to work, because it will have specialized proteins on the surface of the membrane. The structure of the proteins will let this happen, right? Because the proteins, again, structure and function for proteins, they have a structure, they do their job because of the structure. We talked about that in the video on proteins. And so then let's look at the critical steps, right? Where could this go wrong? And really, it could go wrong with almost any of these proteins, right? If you denature any of the proteins, right, that's going to affect this process. Now, when we get to amplification, which I will get in a minute, you'll see maybe even a better way to regulate this, or to put a disruption in this process. Last is regulation, and the regulation could again be, the regulation is the signal molecule, right? Without the signal molecule, none of this happens, right? With the signal molecule, it activates this protein. This protein, maybe the receptor, will go through some conformational change, some shape change that will allow it to start this pathway. Right, so the signal molecule is really our regulator of this process. Now, if we look at amplification here, this is in the phosphorylation cascade. A lot of the times, you're not going to just have something like this, where one molecule creates one response, which creates another response, another response. You'll have a molecule that comes in, it activates this protein, that activates 3 proteins. And each one of these proteins activates 5 proteins. And each one of these activates these 5 proteins. This is very useful because it allows one signal to do a lot of different things. That goes back into fitness, that this helps the organism do things efficiently, right? It doesn't need to send a ton of signals to get a ton of things done. You just need one signal to have a whole bunch of things happen. It also makes it less vulnerable, right? If you mess with one of these, right, one of these yellow diamonds, right, that's not going to mess up the whole process, right? The real critical step here is either with the ligand, either with these original proteins, or whatever, it's the first steps. The first steps are going to be the ones, these are going to be the most critical. Again, not biological terms, but just thinking the logic of it, right? If you're trying to stop this whole process, you want to stop it from the root. So if you're trying to stop a signal transduction pathway, right, the best place to mess with it is at its first steps. You want to denature this protein. You don't want to just have to mess with all these, because there's so many of them, and these could all be different things, right? And that brings us to the last part of amplification. Right now I'm going fast through this. You may want to watch it again if you're not really getting this. But it's a fairly logical process. It's just a chain of events. Right? And I'm going to come back to this question when we go into the immune system video, and we'll actually answer the whole question. I just want to point out this model, because this was on an AP Biology exam, actually the year I took AP Bio. And so what it's saying is essentially you have a bacterial infection that, we'll read it. Some pathogenic bacteria enters cells, replicate, and spread to other cells, causing illness in the host organism. Sounds familiar right now. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways, right? That's what they're doing here. This is sort of a transduction pathway. It's a chain of events, not just with signaling, but kind of signaling inside the cell. Cells normally produce a steady supply of inactive caspase-1 protein in response to intracellular pathogens. Right? Intra means inside the cell. The inactive caspase-1 or caspase-1 is cleaved and forms an active caspase-1. Active caspase-1 can cleave to 2 other proteins. So this is the big part that I want to talk about, the cleaving, right? You have a bacterial infection. This is what is going to start this process, and the active caspase-1 cleaves into two things, right? And what that does is this is a form of amplification, but instead of it being like this, right, where it just has a bunch of things happen, these are all the same, this cleaves into 2, and it causes 2 distinct things to happen. It causes the pores to open up, and it also causes the release of interleukin. Right, and so that's what I wanted to talk about here, is that the amplification doesn't just have to be with the same thing happening. It can be 2 distinct events happening through one protein that sort of splits into 2. Okay, that's it for right now for cell signaling. And again, if you wanted to regulate, or if you wanted to stop this process from going on, what are you going to do? You're going to attack this inactive caspase-1, right? Or active, but mainly inactive. You want to stop this before any of this happens if you're trying to disrupt this process. You don't want to stop the inactive interleukin or the gasdermin. You want to stop it at the source. You want to stop that first step. That's going to be that part that is most vulnerable to being stopped. And again, you're going to have a question on a disruption. So these are definitely things to think about on the effects of disruptions. What happen, and even later in this question, it will ask, what happens if you disrupt the gasdermin, right? Well, if you do that, then it's not going to open up these pores, right? But it won't have any effect on the interleukin, right? So it doesn't stop the whole process if you disrupt it at this step. And so that was actually a question on this, and we'll answer the whole question in the video on the immune system.
I also want to add on to this video a small discussion about feedback loops, right? And so these are going to be important. These might be on the AP exam. It's just another way of regulating, there's specific ways of regulating signal transduction pathways and really regulating a lot of other things in biology. And so there's two main types of feedback loops. We have positive feedback. So this top one is positive. So positive feedback loop. And then this is negative feedback. Negative feedback. Okay, hopefully you can read that. So the main idea, right, behind positive feedback is you want something to keep going, right? That's when you would use a positive feedback loop, when you want something to keep going into some kind of, completion. Right? And so really what's going to happen is something's going to happen. It's going to send a signal. You'll have the signal transduction pathway. It will cause a response. And that response will cause more signals, right? So one signal creates a response, which will then create more signals, which will create more responses, which will create more signals, more responses, etc., etc. So this process, once it happens once, it sets off sort of a chain of events where it keeps on going on more and more. So here's an example of it. I've got the website down here. So when you have a wound, right, you want that wound to heal very quickly, right? You're not trying to make it happen slowly. You want that to happen fast. So that will be a good time to use a positive feedback loop, right? You only need a little bit. The wound tissue releases chemicals, right? So something happens, the wound, it releases chemicals. That's your signal, right? This is not done exactly how I would, but so the chemicals, once they release, they have a signal transduction pathway, right? So chemical signal platelet activation, right? So that's sort of a few steps, right? There's intermediate steps. It's going to be a signal transduction pathway, and then it ends up, right, it signals platelet activation. The response is platelets are activated, right, but the platelets release more chemicals, right, which then releases the chemicals. Those activate more platelets, and those platelets release chemicals, right? And eventually, right, you have a ton of platelets being activated, and those platelets are going to clot your wounds. It's going to heal your wound, right? And so that's the main idea behind positive feedback loops, right? And so usually there will be some kind of ultimate response. Positive feedback loops also get used in the birth, right? When you have a fetus in the womb, then it will, once it starts growing and developing, it releases some chemicals. And those chemicals, as it grows and develops, you want it to keep growing and developing, right? So it releases chemicals that after it grows and develops, after it starts developing, and those chemicals make it develop even more, and so on until you have a baby. Okay. Negative feedback loops are used in a different context. Usually this is when you've got a problem that needs to be fixed, right? But you don't want to overshoot it, right? You don't want, like a wound, right? You want it to heal as fast as possible. So here's an example of this, right? The examples I think are the best for understanding this process. You have an imbalance, so it's not in homeostasis. It's not in that place of balance in that organism. Homeostasis is what an organism wants to be in, right? So in this example, you have high blood glucose levels. You have too much sugar in your blood. Okay, that is something that happens. It releases a signal. It releases insulin, right? Insulin has a whole signal transduction pathway that ends up getting rid of the cell glucose, right? But you don't want your glucose levels to be too low. Low blood sugar is dangerous, just like high blood sugar is dangerous, right? So you don't want this process to keep going on like a positive feedback loop, because then you'll end up with way too low blood sugar, right? And so your cell gets rid of the glucose, your glucose levels are no longer high, right? You're decreasing the signals, or you don't need the signals anymore, so insulin is no longer released, right? And so that's the idea of a negative feedback loop, that your problem's fixed, so it shuts off the process. The process shuts off once the problem is fixed. So these are ways of regulating these signal transduction pathways and regulating a lot of other things in your cell. When we get to gene expression, regulation of gene expression, I'll talk a lot more about negative feedback.